All right. Hi, Max. It's Natalie. How are you?
Good. How are you?
Oh, very good. Thank you so much for joining this Idea Accelerator and Career Conversation. It's really nice to finally meet you. We've been trying to connect for a
Yeah.
bit. I know you were in Boston for a while, but you're not here now. So maybe you can start by introducing where you go to school and what you're doing.
Yeah. So right now I'm in Atlanta at Georgia Tech studying chemical and biomolecular engineering, and then also doing a minor in computer science. And yeah, I'm from the Boston area, but I go to school here, so I'm familiar with both.
Yeah, yeah. Wow. Well, your major and your minor sound like they keep you very busy, and that you are in an awesome place. What do you like about Georgia Tech and the things that you're studying there?
Yeah, so you're definitely right about being busy. There's a lot of work, and yeah, but it's been pretty fulfilling. So it's definitely challenging, but there's also a lot of other opportunities here. So I've been doing undergraduate research, which has been really good, and it wasn't too hard to get into either. So I think that was a benefit of being someplace like Georgia Tech, where there's a lot of opportunities like that. And yeah.
Yeah. That sounds great. So what is your research in? Do you want to give a shout out to your lab PI and
Yeah,
maybe if
sure.
you're working with anyone?
Yeah, so I'm in the Styczynski lab here at Georgia Tech, and it's sort of, there's some chemical engineering people and bioengineering people, and what a lot of them do, or what I do there, is biosensors, so making cell-free biosensors. And yeah, a lot of my work culminated in a paper we published last year, and that was about using a glucose monitor to quantify the readout of a cell-free biosensor. So in our case, we could measure zinc, or the viral load of, like, the amount of a uh, viral load in a blood sample, and then quantify that using the glucose monitor. So
Wow. Well, congratulations on your paper publication. That
thank
is
you.
a major achievement, and that's wonderful. And I think by the example that you gave, the idea of what a biosensor is clear. And then do you want to just say a little bit more about cell-free extracts and why you're working in them versus cells?
Yeah, so cell-free versus, well, I guess starting with what cell-free is instead of using like an entire cell that's sort of doing the operation that you're trying to do in the sensor, you can take the cell extract, which is all of the translation and transcription machinery for the most part, as well as it's kind of crude, so you get a whole bunch of other stuff as well. And then you can use that sort of extract, and if you give it your own DNA, it'll transcribe and translate it and do what you programmed it to do. And since it's no longer part of a cell, it doesn't last forever. It doesn't, uh, grow or divide or anything like that. So it sort of has a limited shelf life with that, but or I guess not shelf life, a limited reaction time. But the shelf life is another interesting part. Is people have figured out that you can freeze dry sulfur reactions and then store them at room temperature. So that's another reason why people might use cell-free versus whole cell sensors, is that you can sort of freeze dry them and then transport them at room temperature and then rehydrate them and use the sensor again, and it still works pretty well. So that was another part of the paper. And yeah, it's super exciting. There's a lot of other cell-free applications, like protein production and stuff like that, because they go a lot faster. They just don't last as long. So yeah, it's a super interesting area for
Yeah,
sure.
I think so. And for biosecurity reasons and all kinds of things, there's a lot of cool stuff. And when you said you put your DNA in it, you don't mean your
Oh,
DNA. You mean the DNA that you're interested in making a protein of, right?
yeah, yeah, exactly.
Yeah, yeah, that's cool. Yeah. So actually for the BioBuilder Club this year, we're going to do an inter-high school study of cell-free extracts and their performance under different
Okay,
conditions.
cool.
So we will tap you for that
Yeah.
expertise as well. That's cool. But I am going to venture a guess that you did not sort of wake up and go to kindergarten on your first day and say, I want to study biosensors and cell-free extracts. You probably had other ideas about, what you wanted to do. Now, were you always interested in science?
Yeah, I'd say I was definitely interested in STEM, like, my throughout my whole life. And yeah, in high school, I definitely got more into science. I really liked my AP biology class
Mm.
and AP chemistry class. So I took those like as soon as I could. And that sort of started me, I guess, down this path. So I did a couple things in high school, but not BioBuilder, unfortunately. And then yeah, in college, as sort of right away, I've been doing this research almost the entire time I've been here. So I started in my second semester, and I knew I wanted to, because I had sort of been peripherally exposed to research, and I knew that was definitely a good way to learn more and experience what it's actually about. So
Yeah.
I reached out right away, and then sort of learned more and more, and liked it even
Yeah.
more as I went along. So
I think that's such great advice because I mean, not research is definitely not for everyone, but I do feel like it is one of the most enticing parts of being in science, or it's something that I do wish we could pull back the curtain on more and invite people in because research, I mean, I'm sure you will tell me if I there are lots of frustrations to it, but it is also a chance to sort of have ownership of a project, to discover something that's not been done before, to do a lot of problem solving. Yeah, so research is also what brought me into science. I found that just to be different than how I was learning science. This feeling like I could actually add to the body of knowledge was really exciting for me. Are there any surprises to you about the research piece?
um, yeah, well, I definitely agree with you on sort of, yeah, I feel like I learn a lot more as well through research rather than like, taking a class or something like that.
Mhm.
Um, and it's really exciting to be able to come up with something new or do something that no one's done before, so no one knows what's going to happen, and then you see the result and learn. And there are a lot of frustrations with that too, for those same reasons, because no one's done it and no one knows what's going to happen. And there's a lot of stuff, especially in biology and with cell free, like I mentioned, the extracts can be pretty crude, so there's a whole bunch of other stuff going on that we just don't know, and it's sort of impossible at this stage to learn all that stuff, like exactly how everything's mechanistically going on. So sometimes, or a lot of times, there's unexpected results
Mhm.
that you can't really explain, and things you think should have happened didn't happen, and you repeat the experiment, but it still messes up the second time. And so there's a lot of stuff like that, just because there's so many moving parts that you don't really see.
Yeah.
But
But
it's
one thing,
definitely
I'm sorry.
exciting.
One
Uh,
thing I love is that, that's where controls come in, right? That's where the
yeah,
whole scientific process comes in, is that, yes, you think you know what you're going to get for, anticipated results. And it's great to try to anticipate what you're going to see. But when it doesn't happen, and it doesn't happen a lot of the time, you
yeah,
have other tools for trying to figure it out. So it's much more of a sleuthing than a
yeah,
recipe, right?
yeah. It's definitely fun. Yeah, failures are also exciting because, yeah, you get the opportunities to sort of investigate and figure out what went wrong. So yeah, those
Yes.
are also really cool.
Yes, especially when you fix them.
Yeah.
Well, that's really cool. So, if you're now doing research as a college student, what would you have liked to have seen in high school, or was there maybe an experience in high school that you thought was sort of pivotal, or a sort of aha moment, or maybe a teacher that helped you reach this point?
Yeah, so I had a really unique experience being near the Boston area. There is a program I did called the Broad Summer Scholars Program. And I think, unfortunately, I think it's just for people uh, in Massachusetts.
Mhm.
But that gave me 6 weeks to work in one of the labs at the Broad Institute, and I got paired with a really awesome mentor who was a PhD student there, and she just had me do some sort of like a cloning project. 6 weeks
Mhm.
wasn't a lot of time, and especially when I didn't know anything. But I that was really exciting for me, and I learned a lot about just the major cloning techniques were super cool to me, like learning about PCR for the first time. And um, yeah, that was it was really exciting. And Gibson assembly was my favorite at the time. Um,
It's still pretty cool. Yeah.
yeah,
Well, I love the Broad Summer Scholars Program. We actually in
yeah,
BioBuilder placed two of our apprentices in their program this past
cool.
summer. they will be thrilled that you gave them a shout out. So, and that it has been such an important part of your experience, because I'm sure they're very proud of you. So that's great. Yeah. And it
Yeah.
sounds like you're interested in giving back and doing additional outreach and teaching. Is that something that you're looking forward to? Or if not that, what is the next thing that you're looking forward to?
Yeah, so I'm definitely interested in giving back. I've been very busy, so I sort of did some volunteering in high school, but since college, that sort of fell off my plate. So I'm definitely looking to get back into that more. Um, and yeah, next steps is a big question for me. So I'm going to be graduating this fall. So this is actually my 5th year. So I will have graduated in 4.5 years.
Wow.
And yeah, there are several reasons for that, but yeah, there's a lot of things I'm thinking about for future next steps, and it's definitely a big question. But yeah, I think the biggest thing for me was just exploring all these different opportunities. I've mentioned research, and I've also done several internships in industry, which have been really eye-opening for me. So sort of get both sides.
Yeah.
And yeah, so
Yeah.
those are big, yeah, very nebulous idea of what's coming next. But
No, I think it's great. As part of these career conversations, we often, with each cycle, we try to talk to a student, but we also talk to somebody who's pursued academics as their career path, and another person who's pursued industry as their career path. And sometimes there's a lot of back and forth. So it doesn't have to
yeah,
be a road of no return. So that's always helpful when it comes time to decision making. Well, I have no doubt that great things are going to be for you, Max, and I sure hope that we can loop you more and more into BioBuilder. That would be awesome.
yeah, for sure.